Okay, everybody, we're going to try and fly through these last two chapters for this unit for you guys. So, gluconeogenesis, chapter 14. So, we're going to outline the path of gluconeogenesis, including the substrates, unique enzymes, and regulatory mechanisms. Review the cycles of glucose and tissue interrelationship when it comes to carbohydrate metabolism. So, remember, when we're talking about gluconeogenesis, gluco meaning glucose, new genesis, creation. So we're creating new glucose from non-carbohydrate sources, okay? It's very important to remember that. So we're going to be looking at stuff like lipids and proteins, converting those into energy sources with the absence of glucose. Okay, so gluconeogenesis is an important metabolic pathway that supplies glucose in emergency situations when the body does not have adequate supply of carbohydrates and runs out of energy. One of the very fundamental concepts is that the body constantly needs glucose for energy production to perform normal cellular functions. Especially the cells of the nervous system and red blood cells cannot survive without glucose. So below a specific threshold level of glucose, the nervous system would dysfunction, and that could even cause coma and lead to death. By definition, it can be said that gluconeogenesis is a process by which the body synthesizes glucose from non-carbohydrate sources. That's what I was just saying there at the beginning of that definition. Although production of glucose via gluconeogenesis is energetically expensive, it does happen to ensure supply of glucose at least to critically important tissues such as the nervous system, red blood cells, and other tissues as well. So our main focus, nervous system and red blood cells, keeping those guys active and fed in order to keep the body alive. So a couple key points. Glucose synthesis is from non-carbohydrates. It meets the needs of the body for glucose during carbohydrate insufficiency, and continual supply of glucose is required as an energy source for the nervous system and red blood cells. There's always a certain basal requirement for glucose. So importance, glucose is the only fuel that supplies energy to muscles in anaerobic conditions, the precursor of milk, sugar, lactose in the mammary gland, and gluconeogenic mechanisms are used to clear products of the metabolism of other tissues from blood too, as lactate formed by muscle and red blood cells, glycerol formed by adipocytes, and propionate formed by beta oxidation of odd-chain fatty acids and by some amino acids such as the branched chain amino acids leucine, isoleucine, and valine. Remember, I live on a branch, L-I-V, live on a branch. And sulfur-containing amino acids such as methionine. Remember what the other one is? What's the other sulfur-containing amino acid? The cysteine. And by digestion of dietary fibers by the gut bacteria. So this is saying that with other metabolic reactions, such as like forming of lactate, glycerol, as well as like the other stuff formed from beta oxidation of these, we end up with certain products that we need to be able to break down and the body needs to be able to use to do something. So gluconeogenesis is going to take those end products, such as the lactate or the glycerol, and take those and plug them through in order to get energy and everything out of them. So that way they don't go to waste, and that way they actually get cleared from the body. So substrates for gluconeogenesis is going to go through quite a few substrates that we can use. So glucogenic amino acids can be converted to pyruvate or oxaloacetate, primarily by transamination reactions, and thus are gluconeogenic precursors. So we have our original amino acids, and we saw that on that chart way back when we were talking about the Krebs cycle and looking at what oxaloacetate can go to and what it can turn into and what malate can go to, if you remember that diagram. We also have pyruvate, obviously because pyruvate is going right into the Krebs cycle. Lactate can be converted to pyruvate by lactate dehydrogenase and serves as gluconeogenic precursor. Glycerol can be converted to glycerol 3-phosphate by glycerol kinase, followed by its NAD+ dependent oxidation to dihydroxyacetone phosphate, DHAP, by glycerol 3-phosphate dehydrogenase. DHAP then enters glycolytic pathway to yield pyruvate. So again, getting all the way back to pyruvate. And we actually saw this reaction. Let's see if I can run up and find it real quick. There was, I believe it was 2 or 3 chapters ago that we talked about these specific reactions. Let's see if I can find it. Here we go. Right in here. See? So we can have the lipid synthesis, and this can go the other way with the glycerol 3-phosphate, the dihydroxyacetone phosphate, the glyceraldehyde 3-phosphate, and then plugging back into glycolysis. And that happens, again, we're talking about in the liver. Okay, let's get back to where we were. Okay. Propionate, obtained primarily from the beta oxidation of odd-chain fatty acids, can be converted to succinyl-CoA and enters the citric acid cycle when it is then converted by a few enzymatic reactions to malate and transported to the cytosol, oxidized to oxaloacetate, and thus serves as a precursor for gluconeogenesis. Others, citric acid cycle intermediates, so all of our intermediates from the Krebs cycle as well can be oxidized to form our oxaloacetate. And this is why earlier, remember, in that chapter I was talking about how if you don't know, guess oxaloacetate, because a lot of this stuff eventually ends up becoming oxaloacetate. So when do we need to perform gluconeogenesis? The fasting or starvation state. That's give or take about 8 hours of not consuming any food. Also a low carbohydrate diet. This is where you get the like the Atkins or the keto diet. The idea is that if your body doesn't have enough carbohydrates, that it's going to start breaking down those fats. Personally, I'd say that not a great methodology about going through it, because then your body develops a bunch of ketones. And ketones down the line, if your body is strictly running on ketones for a long period of time, that can have some negative effects as well. But yeah, it's not really important for this class, but you'll talk a little bit more about that in your clinical nutrition class in a couple of tries. And also intense exercise. We will need gluconeogenesis once we burn up all of our available glucose or glycogen that we have stored. Okay. So tissues and the sites of gluconeogenesis. So in mammals, the liver is the main tissue. Renal cortex a little bit, and then epithelial cells of the small intestine. Just big one is the liver. Liver is the big one to remember on that. Most of this is going to happen inside a liver. A couple of other sources, but places it will happen, but primarily the liver. Okay. Carbohydrate synthesis from simple precursors. We have blood going through all of this. This is just the kind of the chart version of all of these different things, going in and showing where it is all going together into the citric acid cycle, lactate and pyruvate, citric acid cycle, all coming back again to the glucose 6-phosphate. Give me one moment. Okay, so conversion to pyruvate or oxaloacetate is necessary. So any compound that can be converted to either pyruvate or oxaloacetate can be precursors for gluconeogenesis, such as alanine or aspartate. We've got those 2 examples. Conversion of pyruvate or lactate to glucose follows reverse pathways of glycolysis, except there are 3 bypasses. The irreversible and exergonic steps of glycolysis. Remember those rate-limiting steps, the pyruvate kinase, phosphofructokinase 1, and hexokinase. kind of where that phosphorylation happens, and it's highly exergonic. So typically, all of the other reactions can use the same enzyme and go backwards. But these 3 steps are going to need a bypass, something to help them go around it because of how exergonic those steps are. So pyruvate kinase reaction in glycolysis. It is bypassed by the following enzymes in gluconeogenesis: the pyruvate carboxylase and the PEP carboxykinase. These reactions are irreversible, exergonic, and rate limiting. This conversion of pyruvate to phosphoenol pyruvate requires 2 enzymes, that pyruvate carboxylase and PEP carboxykinase. So make sure that you associate these with their appropriate enzymes. So the pyruvate kinase and the pyruvate carboxylase PEP carboxykinase do the reverse of each other. Phosphofructokinase is bypassed by the following enzyme in gluconeogenesis: fructose 1,6-bisphosphatase 1. The hexokinase reaction is reversed or bypassed by glucose 6-phosphatase. All the other enzymes catalyze reactions except those 3 bypasses are reversible. So all the others are reversible, so they don't need any specific help. So this essentially is the exact same process as glycolysis. The only thing you need to remember now is to plug in, rather than like pyruvate kinase, hexokinase, and phosphofructokinase, plug in these 3 or 4 different enzymes in those 3 specific steps. Pyruvate in the mitochondria. So pyruvate produced in the cytosol is transported into the mitochondria, and pyruvate is formed from alanine within the mitochondria by transamination. So like remember alanine is one of our amino acids. Converting alanine directly to pyruvate happens already within the mitochondria, so then we won't need to transport it into the mitochondria. So here we're talking about the first bypass. This again is just diving a little bit deeper into the bypasses that we were talking about. So the pyruvate carboxylase phosphorylates pyruvate and carboxylates it to oxaloacetate in the mitochondria. Biotin is the coenzyme. Note here, 1 ATP molecule is used for this conversion. Acetyl-CoA is a positive effector for this enzyme. Mitochondrial pyruvate is converted to oxaloacetate, catalyzed by pyruvate carboxylase. Oxaloacetate cannot cross the mitochondrial membrane, hence it is reduced to form malate, which can then cross the membrane and is reoxidized to oxaloacetate in the cytosol. Remember talking about that malate shuttle earlier, a couple chapters ago? Similar to that, it gets reduced to malate, which malate can then cross and then gets reoxidizes to oxaloacetate. The mitochondrial PEP carboxylate decarboxylates oxaloacetate to PEP. Another high energy molecule, GTP, is used for the reaction to convert oxaloacetate to phosphoenol pyruvate. So understand that we go from pyruvate to oxaloacetates, then oxaloacetate to phosphoenol pyruvate. So it's almost like we kind of have to go forwards a little bit, getting to oxaloacetate, to then go backwards back to PEP. Now the PEP is converted to fructose 1,6-biphosphate by the normal enzymes. Now we have our second bypass. The fructose biphosphatase 1 catalyzes the hydrolysis of the fructose biphosphate to fructose 6-phosphate. So hopefully you guys remember the reactions of glycolysis, and you really just need to understand and review the bypasses. Again, everything else should be review until we get in our third bypass. The glucose 6-phosphate hydrolyzes glucose 6-phosphatase hydrolyzes G6P to glucose and an inorganic phosphate. So gluconeogenesis is energetically expensive but essential. So in order to produce a molecule of glucose from pyruvate, we need 4 ATP, 2 GTP, and 2 NADH. comparing it to glycolysis, we only need 2 ATP molecules. So gluconeogenesis is really, it's a much more expensive. We're essentially almost using 6 ATP, 6 chunks of energy in order to create a glucose molecule. But it's necessary because, again, if we don't have glucose, we need to be able to create glucose to have it go to the parts of the body that need it. So that's why the liver is in charge of this, and then the liver sends it through the bloodstream, and it will go to the nervous system and the red blood cells and everywhere that needs it in order to maintain those tissues. So glycolysis and gluconeogenesis are reciprocally regulated in the liver. So reciprocal regulation occurs when the same molecule or treatment has opposite effects on catabolic and anabolic pathways. Reciprocal regulation is important when anabolic and corresponding catabolic pathways are occurring in the same cellular location. Okay, so in this case, fructose 2,6-bisphosphate is going to be our reciprocal regulator. So reciprocal regulation prevents wasteful operations of both pathways at the same time. So some of these other important concepts to know: in well-fed condition, the levels of insulin is high and glucagon is low. This increases levels of the fructose 2,6-bisphosphate, favoring glycolysis. The increase in that levels increases phosphofructokinase 1 catalyzed reaction, which in turn activates pyruvate kinase reaction in a feed-forward manner, and thereby increases glycolysis and decreases gluconeogenesis. So in contrast, in the fasting or starved condition, the levels of insulin are low and glucagon is high. This condition decreases PFK1 activity, which in turn decreases pyruvate kinase activity and increases gluconeogenesis to maintain glucose homeostasis, and glycolysis is decreased in the fasting or the starved condition. So that's really all that's there for this chapter, not a whole bunch. Main thing, focus on these bypasses, review your glycolysis, as well as review specifically those enzymes, which are the irreversible exergonic steps of glycolysis, and then how we bypass them, as well as take a look at those major substrates, what it is that we're putting in order to get the glucose that we need. Okay, good luck on your studies. Remember to work hard, study hard, and have fun.